Alright, welcome guys, I am Cody Levitt, and I'm going to be teaching you a little about a couple different things. First we're going to start off with homeostasis, then we're going to move into transport, and then lastly we're going to talk about osmolarity and tonicity. This is mainly for the BYU Idaho 5461 class, but if anybody else wants to join in and watch, you're more than welcome to. So first, homeostasis. What is homeostasis? Homeostasis is the ability, forgive my spelling, homeostasis is the ability of the human body to maintain an environment internally that will help it function properly. And so I'm going to draw out a graph to represent this. So we kind of have a range. This is called, just your normal range. You have a set point. That set point is kind of where you start. And so we're going to talk about we're going to talk about a feedback loop. All right, so we're going to be talking about feedback loops as it affects homeostasis. So a feedback loop in an ideal circumstance is a loop. So we have a stimulus, which then goes to the receptor. The receptor sends information to the control center, and the control center will then send a message to the effector. So in an ideal circumstance, we want this stimulus to make it all the way from the effector back to the receptor to tell our body that everything is good, that everything is working fine. So these different pathways, this is the afferent pathway. And this is the efferent pathway. So this loop, when it comes from the effector and goes all the way back to the receptor, this is called negative feedback. So we'll turn back to our graph over here. So a good example of this, if I was to drink this Baja Blast, this has 73 grams of added sugar. My blood sugar is set at a normal range right here, where it goes up and down based on, the meals you last had in this set point. So if I was to drink some of this, my blood sugar would shoot up, probably outside of normal range. 73 grams of added sugar is quite a bit. And over the period of time, it would come back and it would start fluctuating again until I ate another meal. And this is ideally how we want our bodies to work. And so the definition of negative feedback marker's not working that well, is a return to set point. And so the definition of positive feedback is when we don't return to that set point. So going away from the set point. So let's just real quick redraw it. Kind of give an example of positive feedback. Okay, so a good example of positive feedback would be somebody in a car wreck. So they start losing blood So our blood volume is going down. So what we would expect is that our blood pressure goes up, so our body tries to compensate for it by increasing the heart rate, and then our blood vessels contracting. Peripheral resistance goes up, and so we'd expect these to continually rise to try to compensate for that low blood pressure. But when we lose so much blood that this continually goes until we just keep losing blood, it eventually fails, which results in the blood volume down, blood pressure is down, our heart rate is down. So we're trying to just savor that little last part of blood, and eventually it fails, and that's essentially why people die. And so during this process, so we're going to see this as just your blood pressure is what this is represented. So it starts good, kind of drops a little bit, goes up, and stays within that normal range until it eventually can't, and then it just keeps going out of the normal range and starts failing. So this is what positive feedback. This is a really good definition of what positive feedback is. And so we're going to next move on to our next. All right. So now we're going to talk about transport. Transport is important because it's talking all about how things are going into and out of the cell. So there's 4 different types of transport we're going to talk about today. First one is simple. We have facilitated. And then we have primary and secondary active transport. Okay, and we're going to kind of make a table just to summarize all of these principles. So, first use is, does it use ATP? What is its relationship with the concentration gradient? Does it use transport proteins? What does it do when it gets saturated? And is it selective? Okay, I'm going to switch colors here. Green marker. Okay, simple. Simple does not use ATP, neither does facilitated. But primary active transport, ATP is the key there. That's pretty much the biggest relationship, so yes. And secondary active transport. So secondary active transport depends heavily on primary active transport because it uses the same molecules that go into the cell. So this is indirectly affected by ATP. So gradient, this is vital in the simple. So you use concentration. It goes, your concentration gradient is super important in simple transportation. Facilitated, this uses your concentration gradient as well as the electrical gradient of the cell. Primary active transport, this actually goes against the concentration gradient. That's the main use of this ATP, is that there's enough energy to go against the gradient. Secondary active transport, there's actually 2 types, and so your types kind of depend. Here, so we have symport, and we have antiport. So symport, I'll draw out a little bit. Draw over here. That's our protein here. So symport two more molecules that are coming through, and they're both coming through at the same time, going the same way. Antiport, this word, prefix anti, is against. Great drawing. Amazing. Just kidding. Okay, and so we have two different molecules. So this one is inside the cell, this one's outside the cell, and they kind of pass each other on the way. The idea is using the energy from one to propel the other one. And so in symport, it can go with or against, and antiport will always be with and against, because against, I can spell, that makes that easier. Okay, perfect. Transport proteins now. So simple is the only one that does not use a transport protein. All the rest of these are yes. Transport proteins help them to carry out the job and just allow it to get through. The same trend is found here in saturation. Simple doesn't actually care about that. It just stays linear. So yes. So what I'm talking about when I say that, I'll draw a little bit of a graph here. And so saturation, when you think about saturation, it's all about like, a high dense of something. So saturation, when we have, well, a good example is, I'm not going to talk about that. I'm not going to go on that rabbit hole. Anyways, saturation, when we just get so much of one molecule that it starts just becoming too much. So this graph is going to be right in concentration. So these are affected by saturation. So it's going to start off linear, and it's just going to keep going, and we hit a point where we start slowing down, and it just kind of plateaus off. That's supposed to be straight, so that looks like it's going down. That plateaus off because we don't have enough room in the end. There's not enough space for all of these extra molecules, and so our rate is just going to plateau off. Where in simple transportation, it is just going to keep going. It's just linear. It stays constant. It doesn't accept too much at one time. It just stays linear. And it keeps going. And I think part of that too is to do with this next portion. Simple is not selective. When we talk about selective, we mean very specific. All the rest of these are highly selective. Simple diffusion is not selective. The rest of these, facilitated is yes, primary yes, and secondary yes. So now we're going to go on to our next portion, our final portion, talking about osmolarity. Okay. All right, everybody. Sorry I had to go do some stuff. The camera probably moved. But so now we're going to talk about osmolarity and tonicity. So osmolarity, look at this. So this is a solution. We'll change the colors here. As an osmolarity, you look at these and they have the exact same concentration of a certain salt or mineral or whatever it may be. So these are the same. We call that isoosmolaric. And so you look at them and you're like, oh, they're the same, but they're different things. So they have the same concentration, But when you put them together, this green is permeable. That means it can go across the gradient. So we're going to leave this here. It's going to go across. But the blue isn't able to move across the gradient. This only allows the green to come through here. And so then we would refer to this side as hyperosmotic. I'm sorry. This is what tonicity is. This would be hypertonic. Because hypertonic is all about this moving of particles across the gradient. And so we're going to talk about this within the cell. I'll give you a couple different examples. I really enjoyed this unit because this is a very applicable thing to all medicine. And so we have a cell. 2 points in, 2 points out. They're the same. This would be isotonic. It would also be isosmotic. But there's a few different solutions that we can use and that they use in medicine. One of them is NaCl. This one is dextrose. I'm just going to go through these two and the different situations we can make. And so, when we have something, okay, these two dots in here. Say we put dextrose in here. Dextrose just has these 2 points, but dextrose always goes into the cell. But then when it goes into the cell, it metabolizes. Well, it's almost like this just disappears. What it does for the cell, though, is it floods the cell with water. Yeah, I guess we'll leave this in here. I'll draw another one real quick. So this is the after. That's before. No, I mean, it looks completely different, but what happens is all of this fills with water because water goes to where the minerals are. And what it does is it swells the cell and then it becomes hypotonic. It's still isosmotic, but it's hypotonic because of what happens with all that dextrose. Then we're going to look at what happens to NaCl. NaCl is kind of a little bit of a different story here. So they were also sitting. So we have that NaCl. And NaCl, it can't go across the membrane of the cell. It's non-permeable, so it's not allowed to go across. So this becomes because there's more concentration outside the cell than there is inside. And so this would cause the water to flow out and causing the cell to shrink. But in this circumstance, the hypotonic causes the cell to swell. Either one of these are very, they're not ideal circumstances for a cell. We want our cell to sit in an isotonic position, but when our body's not working correctly, that's when that saline those different types of solutions can be applied in a hospital setting to help our cells either, you know, fill up more, hydrate more, or dehydrate by shrinking. And so that's kind of where those are applied. And yeah, that's about all we have to talk about with osmolarity and tonicity. Thank you for watching this video. Appreciate you guys.